Vado?
Good morning, good afternoon, everybody. So we are going to start the keynote. We are in the last part of this meeting that was very intense, very interactive. And now it is my very pleasure to introduce you the talk of Professor Diego Mantovani from Laval University. He woke up very early to be in this session. I would really like you to enjoy the speech of Professor Mantovani. Then I'm very happy to have him in the conference. Thank you, Diego. You can have your time now.
Perfect. Thank you very much, Serena, for a very much appreciated invitation and for hosting a dual conference on site and in video conference. I am particularly unhappy to do not be able to be in Pisa with you, but the Canadian frontier are closed, so we cannot travel, and it's very difficult if we go out then to come back. So it's my honor to discuss with you today a project that is just completed, and I think it's very interesting in the framework of this conference. So I plan to entertain you for some 30 minutes. So now is 8:16, let's say up to 8:45, in order to leave some minutes for discussions and eventually comments, ideas, and others. So in the next 30 minutes, I wanted to bring you into the field of plasma-based surface modification for bioactive molecules grafting for regenerative medicine. This is an international project run in collaboration with Abbott and with Belgium and France partners, and a lot of thanks, but mainly to the student involved and to the partners and to the companies. And let's go straight. And 1 person on 3 in the world die by cardiovascular disease. So this is a natural artery, and I remember when I was a student in Milan, we got a lecture on how beautiful is this mosaic. So this endothelial cells layer, monolayer, completely confluent, very aligned and oriented with the blood flow. Unhappily, aging will bring deterioration and narrowing and the arrival of atherosclerotic plaques. Atherosclerotic plaques mainly are accumulation inside between the intima and the media, so in the second and third layer of the arteries, of metabolites and especially catabolites. This will induce a rigidification of the arteries and will bring to the requirement of pharmacological treatment and later on eventual surgical treatment. It's something that affects roughly 300,000 replacement per year worldwide. So the media is the muscular part of the artery. The intimal is, let's say, the moquette and the carpet of the arteries, while the adventitia has more an anchoring role. So in our lab, we address a transversal and converging approach to work from different points of view, including biodegradable metals. Our dream for this is to go and correct congenital diseases in the newborn babies, even before born, in order to correct, for example, a vascular or cardiac defect, in order to limiting the progression of atherosclerotic or our problems. We work on regeneration, so creating models of vessels and cardiac patches that can be used for validating materials and surfaces that we engineer in our lab. So probably most of you know what is and how it works, the implantation of a stent, and basically the catheter is inserted inside the artery and there is a stent inflating. You can see here the 3 different steps of inflation, and finally catheter is removed and the stent remain in place. This project, what I want to present to you, concerns stent. 300,000 of stent are implanted every year in North America, and roughly 3 million of stent are implanted worldwide. $1,000 each is a 3 billion market. A part of the market consider that 20 to 30% of stents would reach a complication within 6 to 12 months of implantation. So basically means that 1 stent or 3, if possible, could be retrieved. So implantation of stent is not a completely new technology. Actually, is quite, now we can start to say, quite old. The pioneering studies in 1974, where the first peripheral human balloon angioplasty was performed, and initiated the idea of correcting atherosclerotic problems from inside and not with conventional surgery. We needed to wait 10-15 years in order to have the first coronary stent implanted, and it's only since the early 2000s that stents now are routinely implanted. Cardiac and vascular stenting is nowadays a very well accepted surgical operations, and the main interest is that is not run in conventional surgery but intra-arterial, so intravenous, intravasculars. However, as I mentioned to you, there are complications. So basically 1 stent or 3, and in some cases 1 stent on 2, reveal some major injuries at the endothelium after the implantation. So there is an inflammatory response to the penetration of the structure inside the wall. As you can see here, the stent, when implanted, will not remain only, let's say, deposited to the intimal layer, but it will enter and break part of the intima and reach, in general, the media and even sometimes the adventitia. So there is an uncontrolled proliferation of smooth muscle cells called in-stent restenosis. Strategies to decrease this restenosis rate are necessary. I can add, and there is no visual support to that 10 years ago people imagined that just dipping the stent into a drugs would have increased the clinical performance rate. But this was not true. The fact of binding to the structures antistenotic molecules like paclitaxel and sirolimus were just, let's say, big dreams. Today we know that apart a few cases in a very degenerate vascular bed, so this means very aged patient with a lot of complications, and several surgery already underwent, in general, we cannot count on drug delivery a drug-eluting stent for, let's say, conventional surgeries. So companies, and Abbott is quite innovating in this, decided to explore what happens if we decrease the mass of the stent. So going thinner has become in the last 5 years the main targeted approach. And this is also very easy to see in this image where you see the section of the stent really going down. The last chrome cobaltum or zero stent is 60 microns in structures. So this means if you go down with in dimension, in geometry, this means you need to increase the mechanical properties of the stents. But today, generally, stents are implanted much less than 140 microns initially planned, and chrome cobaltum is replacing slowly the stainless steel as a gold standard for operation. However, even with a reduced strut thickness, the restenosis rate decreased from 25 to 15%. So this means that one stent of C6, one stent of 7 still have a problem. There is also the potential release of cancerogenic ions, nickel and chromium. Chrome cobaltum is an alloy with 30 to 40% of chromium. So drug-eluting stent became a reality, let's say, around 2005, sorry. But in 2015, was evident that major complication like late instant restenosis arrives. In other words, the fact of releasing for years paclitaxel, sirolimus, and derivatives compromise the homeostasis of the entire organisms and resulted in an unbalanced blood coagulation cascade. So the critical compromise in endothelium recovery was due to non-specific drug, and the idea is how can we improve the performance of the devices. This project results from a joint connection between cardiac surgeons in Paris, Abbott Research Division in US, our lab, and few other labs in Paris and Belgium. And the idea was brought mainly by our cardiac surgeons. The idea is, let's work on the equilibrium between materials and biology. So surface modification, we know it's a proven, interesting approach to modify the first layer of atoms without touching, disturbing, affecting the bulk properties. For example, you can imagine on a surface, and generally this is done with polymers, to functionalize the surface, so change the wettability and finally affect the surface finishing. So the clinicians brought these 3 blocks. We need a cardiovascular devices. The future stent should be a stent anti-inflammatory, pro-endothelialization, and antithrombotic. So our idea, our solution was why to do not graft bioactive molecules to enhance the device integration. This is not very much different from what, for example, our colleagues presented early this morning. And very schematically, our approach was to take L605, so chrome cobaltum, medical grade, used for fabrication of millions of stents around the globe in the last years, and coat a deposition and then functionalize the coatings, and finally revealing cracks in the coatings when the material is deployed. So in this project, one of the innovative approach was to challenge the use of a polymeric layer. If you took, for example, Abbott, they have on the market X stent. This is a stent just coated with a very thin fluorocarbon layer to avoid adhesion and proliferation of smooth muscle cells. In this work, we wanted to bypass, to limit the coating presence. So our challenge was, can we graft directly a functional group without acid, without chemical liquid, humid chemistry? How can we attach to a metal a molecules, for example, a very small peptides, just in order to get a strong binding to resist the deployment in the body. Our approach was to explore the direct functionalization by plasma. And this is something that resulted very original. No one ever published, reported a work on direct functionalization. Direct means take the metals, put in a plasma reactors, and anchors create functionalization groups by plasma treatment. So depending from the gas that you use in the plasma, you can fix to carboxyl, to amine groups, or other groups. So functional groups are to be used as an anchoring point for biomolecule grafting. In our case, in the following of my presentation, I will mainly focus on amine groups. So when you have a surface with amine group and you want to attach bioactive molecules, in few moments I will tell you which was the bright idea brought by clinicians for the new bioactive molecules, then you need some different strategies, but mainly linking arms. So in general, you can see works in the literature with anchoring to functional group DNA, heparin, hyaluronic acid, enzyme, proteins, antibody. And in general, linking arm are used because you can modify the performance of the bioactive molecules. Depending from the availability of the molecules to the biological surrounding environment during the implantation, then you can have different results. In this work, we explored two linking arm, one PEG, so 1200 carbon chains, and glutaric anhydride, 6 carbon chains. The idea was really to let the molecules to be available to the bloodstream, to the cells colonization, and to extracellular matrix proteins interactions. What I present in this slide is mainly came from clinicians' point of view. They selected a biomolecule of interest, which is in the family of PCAM. These are very, very platelet endothelial cells adhesion molecules, or the cluster differentiation 31. And the idea was really to focus on these molecules that are very small because they are 6 to 12 amino acid sequence, and they have I wanted to say tremendous, but then I voted to Donald Trump and I wanted to avoid the use of tremendous. So let's say a very, very interesting adhesive proteins like endothelial cells and platelet specific. So the idea of this PCAM1 was to inhibit the platelet activity and aggregation, and finally the clinician in Hôpital Bichat in north of Paris patented a synthetic short chains that they invented, and the name is P8RI. So it's a small sequence of 8 amino acids supposed to be responsible for attracting endothelial cells very, very rapidly to the surface. So by grafting these very short peptides, we expect to generate a biomimetic surface to improve chrome cobaltium biomedical stent performances. So the idea that we developed was take the stent, modify in plasma. So we had a company developing a very interesting plasma reactors in which you can treat up to 18 stents in the same time, like a revolver bullet support, and in presence of nitrogen containing gas in order to create amine group on the surface. To be honest with you, here between the oxide layer and the engineer, we need to engineer the oxide layer. I will explain in a moment how. And finally, we graft the biomolecules. So for stent applications, the idea was to be corrosion resistance, deformation resistance, and chemical controlled composition. The plasma amination efficiency was the criteria for plasma treatment success, and finally the feasibility to graft molecules with different linking arm, expecting different biological performances, are the 3 targets expected from the work. So about the feasibility of the approach, this was published a couple of years ago, and what is interesting in that this is, to the best of our knowledge, the first time that a metal was successfully functionalized in a plasma reactors. Plasma are generally mainly devoted, especially low vacuum plasma, are devoted mainly to polymers. Finally, surface optimization, and basically this is optimization of the linking arm, and finally grafting work now published in biointerfaces. And so the methodology was the following. I go fast. If you are interested, I can share results with you very easily. And the idea was start with a cleaned stent, electropolish, and in order to engineer a bit the oxide layer, and finally to graft amine groups. Amine groups explore the PEG and the peptide, and then assess the biological performances. The peptide, this P8RI, was patented by a company that now is working on clinical level on this project. XPS and high resolution immunostaining fluorescence were used to assess and to validate the protocol. So why polishing? I make you a very short history here, but this was a 2 years research carried out in a teamwork in order to understand, should we remove completely the oxide layer, or how much should we leave of the oxide layer, and which will be the best roughness? One should know that the stents for cardiac, vascular, and neurological application, generally they are surface treated in order to get roughness of 4 to 6 nanometers. So the plasma reactor is this one. So the proof of feasibility was done in a conventional plasma reactor, and then a more, let's say, targeted plasma reactor was developed with 18 stent can be positioned inside. Amination was done with a different condition, and basically in a post-treatment, and the plasma optimization arrived to a value of 10 to 20% of nitrogen, which results in 1 around 1.5% of amine group. This is more than enough. Don't think that we need 100% of amine group on the surface. So the homogeneity of the stent functionalization was assessed. So the top part, the bottom part, and the central part, and basically this was confirmed. We used the bromium in order to derivatize the amine group presence, and so to be able to see, to assess the aminization capacity of the process. And finally, two different linking arms were used for connecting the peptide graft. So this is the PEG long chain, and the glutaratic acid was second. So in general, no nitrogen after electropolishing. This means that the nitrogen on surface come mainly from the plasma treatment. And finally, the PEG is attached, and the PEG peptide is there. This is an interesting table because you can see the evolution, for example, of what you bring to the surface, and you can notice what that oxygen and carbon presence increase during the PEG grafting and the peptide grafting. What is important is to see that basically 5% of nitrogen was the targeted approach. And then some in vitro test in order to assess in the lab, and especially with endothelial cells, was carried out. So the phenotype, so multiplex analysis of sub-inflammatory factors, and some distribution of endothelial cells by immunocytofluorescence was carried out. So this is the short chain or with glutaratic acid. This is the long chain linking arm with PEG. I think you already captured the potential of both. And these are the in vitro results of endothelial cells phenotypes. So basically the vascular cells adhesion molecule VCAM1 decreased, and this means that mediate the adhesion of platelet to endothelium, which is very positive for us. Interleukin 6 decreased. This means that basically the surface is less inflammatory, and the tissue factor pathway inhibitors TFPI increased. This is a coagulation inhibitor. These are three factors quite considering from cardiac surgeons. So in general, the surface after the grafting is more anti-inflammatory and anti-thrombotic than electropolish. So there is a synergistic effect between the PEG and P8RI. Basically, I suspected that before doing the experiment, our expectation was that the more freedom was given to the peptide, higher would be the biological performances. This is a result of electropolish. This means no PEG, no peptide. This is PEG coating and PEG and peptide. We are looking at endothelial cells 1, 3, and 7 days. And basically, the P8RI grafting increases endothelial cells viability and the anastomotic integrity. And then we transfer this to a commercial cardiovascular device. So 80 stents was provided by Abbott, which was a good partner in this project, and then started to be a bit complexified, because what I presented to you up to now was done mainly on flat surfaces. All the processes were optimized on flat surfaces. And now you have in the hands an 80 micron structure stent, 3 centimeter longs and 3 millimeter in diameters, and when expanded is maybe 3.5, so 1.7 to 3.5. So a very teeny devices, very complicated to manipulate. Can you imagine to do XPS? Can you imagine to do contact angle, surfaces, profilometry? Everything become more complexified. And when the challenge become high, you need to use very advanced techniques. So we went to TOF SIMS imaging, and this is an image obtained on 500 square micrometer square and 150, and what you see in yellow is basically the presence of P8RI on the stent structures, because actually Abbott wanted to be convinced that the peptide was really grafted. So TOF SIMS analysis confirmed the presence of a peptide by detection of the lysine fragment C4H8N plus positive ions. And TOF SIMS, imaging is not something easy to carry out, even on flat surfaces, but imagine here on 80 microns structures. This is a positive and negative mode for TOF SIMS imaging of a commercial stent modified by plasma-based strategy, and you can see in general how clear is the yellow segment show the presence of a peptide. And then we went in vivo, so different in a pig model, and we compared 3 stents. Multilink, which is a bare metal stent, nothing attached. X-Science, which is a drug-eluting stent coating with fluorocarbon and grafting with sirolimus, and finally a PEG PA tire. This was a very interesting approach, and I wanted to spend a minute on this. When you test a new strategy like the PEG P8RI grafting molecules without acid and no wet chemistry, then you need really to find the positive control and negative control. And while basically this is something that it's really, a clinicians will not test a new device, a new strategy, without comparing with what exists and what they use, and basically what they know it work well and what they know it work less well. So that's why bare metal stent today for a middle-aged patient is considered as the gold standard for a rapid endothelialization. Drug-eluting stent is, for the same patient, is considered a negative controls. No stenosis within 6 months, but after 10 years and completely unbalanced hemostasis. So that's why comparing with a bare metal stent that do not have any peptides, any molecules, any delivery, and drug-eluting stent that, yes, there is a drug releasing, but in the same time we know that at medium term, 5 to 10 years, complication will arise. So we were implanted for 7 to 28 days, and mainly because the idea of PA tire, P8RI, is really to check the rapid endothelialization boosting effect that is provided by the presence of the peptide. This is the stent once implanted in pig, and this is during, let's say, explantation, and finally we collect materials to go further analysis. And this is the very interesting results. You have a bare metal stent, you have drug-eluting stent, and here you have the peptide grafted stent. So let's look, this is a endothelialization at 7 days, and this is an OCT visual an image of the stented artery and how patent it remains right after. So clinicians was very, very interesting because of the very high endothelialization stent compared to drug-eluting stent. Drug-eluting stents, we know that endothelialization is not fast, but in bare metal stent is supposed to be fast, but there are some, let's say, coagulation, the presence of coagulation. Here on the peptide, you just see endothelial cells and basically some platelet, but not activated. Here they are not spread. And the presence of the eluting drugs, paclitaxel, show that here too. So the peptide generate the same answers, response than a drug-eluting stent, but with a rapid endothelialization that generally is observed with bare metal stent. This is an OCT image where you see the patency, and basically you see that for drug-eluting stent is not bad at all, but the best is really for the peptide-coated stents. So there is a less in-stent restenosis compared to bare metal stent. And this is the in vivo test 7 days after, and you can see bare metal stent, so basically no coagulation, some coagulation here in drug-eluting stent, and finally plasma peptide coated, quite clean. So in conclusion, surface modification can improve the biological performances without compromising the material properties. The best amination efficiency were observed with maintaining of corrosion and deformation resistance. This developed a new platform for the direct functionalization of metallic substrate, and developed in flat was transferred to commercial cardiovascular devices and stent. This plasma-based strategy, when coupled with CD31 peptides, improve both in vivo biological performances. And when compared to commercial cardiovascular device, this peptide strategy include a higher endothelialization than drug-eluting stents and antithrombotic activity, especially if compared to bare metal stent. And finally, a decrease in the stent restenosis when compared to bare metal stent. Today, this project is under development, and Abbott invested in a small spin-off with today 12 people working on it and carrying out longer animal studies and starting the preclinical analysis. So we have a good expectation that in 2-3 years, this project and these results will arrive to a product on the market. And a number of activity transversally are ongoing, including the transferring of these peptide techniques, grafting to balloon angioplasty, for example, or other metallic devices. Two of our past PhD students now are involved in the companies, and this is also, let's say, an interesting results for professors like me. And clinicians are very excited to have been introduced to TOF SIMS and this kind of analysis, while companies are generally really prudent with this kind of work and project. And this is why I want to emphasize that this work was not funded by Abbott, because we wanted to keep intellectual properties clues, and especially freedom in analyzing whatever we want. And this was a French ANR. This is a strong Canadian health research funding and an European grant was associated, in which Abbott played a major role in justifying the future of the work, but and of the development. So I wanted also to provide to you this, let's say, transferring to real products, let's say, image of what the project is becoming right now. I really appreciate again your invitation, and I hope I was not too much boring. And I am very open for a question, discussion, ideas, or comment. And it was a pleasure to be here with you today, even if in remote. Thank you very much.
Thank you very much, Diego. This is a very remarkable work. I think this is still an unsolved problem. So it's very nice that you could combine the molecule directly to the surface of the metal. So I think that the approach, is very immediate, very simple, and the translation into the clinic is also maybe faster, no? Anyway, I have a question. I was wondering, for example, so when you attach a peptide to your surface, is it a medical device or it becomes more complexly regulated device?
This is a very, very interesting question. From a regulatory point of view, the fact of introducing, even if it is a short sequence of peptide, complexify the regulatory procedures. Basically, these need to be assessed with a trial in clinics, and you need to demonstrate and comparing with bare metal stent the superiority of the materials, or at least equals, so the peptide efficiency. But also you need to convince the regulatory bodies that the peptide will not interfere with any other biological cells and molecules and tissue and fluids in the blood. So there is quite a lot of in vitro and in vivo study. I could not present everything, but potential side effect of the peptides needed to be assessed. And basically this is also the stability of a peptide needed to be assessed. I couldn't report here because it's under confidentiality with Abbott and the company in Paris. But what is interesting to note is assessing the stability of the peptide require to explore the presence of a number of enzymes in the environment, especially during an inflammatory process like the post-implantations. So it make no easy. I think I partially answer to a question that pop up in my screen about the stability. And yes, this is something absolutely not trivial. Being the process ongoing, it's difficult to be precise and to tell you exactly which are the enzymes that we analyze, but so far it looks that the 8 peptides are easily degraded after 2 to 6 weeks of implantation. And this is interesting because the idea behind the project from a clinical perspective was really to get faster you get the endothelialization, lower will be the potential complications that will pop up in the stent. Thank you very much. It was a very interesting question.
The answer. There are many questions in my mind. I don't know if there is any more from the chat or okay, so this one was from Nihal.
Exactly. I think I partially answered with the peptide in vivo and the enzyme. Sure, there are enzymes that can degrade the peptides. The fact that the peptide is binded with the PEG also could generate that the PEG link can be is stable, but it's not eternal, so it can be degraded too. So yes, there are enzymes that can degrade, but as I said, the idea is to keep the peptide at least 2 weeks attached to the stand.
Have Ipshita also another question?
Sure, Ipsita is very smart and she look rightly into the point. Actually, you completely right. So the answer is yes and no. So this means no, I didn't compare smooth muscle cells proliferation and attachment with and without peptides, but mainly because it's not it would have required to work in vitro on a separate topic, and when you work with companies they really want to bring you, let's say, focused. But this is something that we are planning to do. Actually, instead of when you work in R&D and I, research and development and innovation project with a company, they only work that you focus on improve. You must be positive. They are not interested in understanding why it will not work. So the reason for which smooth muscle cells cannot interact with the peptides is not their interest, but it's very pertinent in the project. So I completely agree with you. This is a very interesting questions, but no, I don't have answer right now.
Okay. Thank you. Also, we have a question from the floor. We have Paolo Scopesce. So he has a company with plasma treatment. So we'll be maybe very interested.
Hi, so very interesting talk. Yeah, just only one question about of course, the possibility to treat the metal directly with the plasma. So I know from literature, some people are studying the possibility to provide some PEG-like layer directly with plasma. I know if in your application, if your case makes sense, or if you already tried this kind of plasma deposition. Thank you.
We already tried to develop a PEG layer on metals, but do you know, PEG, I really like PEG as a linking arms because as a provide a huge mobility to the molecule that you bind on the top of a linking arm. But the idea of fully coating a PEG, I think in terms of wettability, I am not sure it's interesting because it becomes very, very attractive for any kind of biological molecules. While it can be very interesting for contact lens, for example, because these improve the supportability, the ability, the easiness to which a patient can have them. I am not sure that for stent in bloodstream and in and on endothelial surfaces in presence of a rich extracellular matrix and 2 layers of cells, this is interesting. I never tried to, I already tried to coat metals and polymers with plasma-based treatment and PEGs grafting, but I never tried to assess how rich can be the interaction between a peg coating materials, metals, with extracellular matrix proteins. I'm not scared, but I think it will be difficult to assess what the peg will generate in terms of if I can use disturbing the or unbalance the homeostasis, the local homeostasis of cell cultures, the presence of 2-3 layers of cells and their extracellular matrix. It's something absolutely to test. I am very open for collaboration, and this project is finished, so no problem. Great. Thank you very much. I hope I answer.
Thank you. If there is no more question, I will ask my last question.
Please.
Okay. So I know there are other types of stents, not only for the coronary. For example, the tubaric stents for the Eustachian tube, and also for urinary tubes. Do you think your technology is flexible? I think so, but what do you think? To attach different molecules for different like epithelial cells or other cells?
This is a very interesting question. So the idea to use plasma treatment in the range of 200, 300, 400 watt and treatment long 15 to 20 minutes in partial 10-5 torr vacuum, I think it's something easily transferable to a number of materials, for sure, including ceramics, composites, and even decellularized matrix. But the idea of transferring to other applications, I know for sure that some dental applications are interesting, especially for growing of a gingiva on a part of the implants and not on the other parts to avoid bacterial retention and so on. It's something actually expected. So we already tried to work a bit in dentistry. Urinary tracts, this is not limited to stent. The sense that any kind of surface, if you have reactors where the implant can enter, one of the nice things of the plasma is that it's really symmetric. So it's you're dealing with homogeneity of complex of the treatment for complex geometries. I think it's something that plasma can show very very high and positive effects on it. So yes, I think it's definitively possible. No, I didn't try to apply this to stent used in other application that coronary, cardiac, and neurological. We are working quite a bit on the next generation of stent, ultra thin, 30 microns, and I didn't try in others. But yes, stents are even used in esophageal, even in pulmonary. There are a number of, let's say, less numerous, but quite adventurous applications. Thank you, Serena.
Exactly. Thank you very much, Diego. So I think so. If I receive other questions in the chat, I will forward them to you. So I thank you very much for your time spent here. So we are waiting to see you in person, Diego. So you keep the morale of any conference, even during the COVID, very, very nicely. So we expect to meet for in the next, maybe next year, hopefully.
Thank you very much, and I really hope to. And thank you for your time and for the quality of the presence of the people. I really appreciate. Thank you.
For the compliment. Thank you, Diego. So if you want, you can still follow. You can still stay if you are interested in the next session. You can stay, or you can go and follow with the YouTube as you prefer.
Perfect. Thank you very much. Bye-bye, everyone.
Okay, bye-bye. So I want just to remind that now we're going into separate sessions, and since there was a delay in the previous one, so the presentation of Bahare Azimi is postponed as the last. So we will continue in her session in room B by adding the presentation in the last. Thank you very much. So now we can go with Francisco Martinez, Martin Martinez. Thank you, Fran, for being here.
Hello.
And we start with the in silico. So now computational is becoming very important in the material science because it has a lot of advantages comparing to experimental. So also we can combine the two and have like faster results and much more reliable. So that is why I wanted to have an in vitro session, in silico session. And I'm very glad to have here Dr. Martin Martinez that moved from MIT to Wales in the UK. And I leave you to his presentation. Thank you, Fran.
Thank you. Thank you, Serena. Let me share the screen.
Yes.
All right. You can see my screen, right? Yeah. Okay. So thank you, Serena. I'm very happy actually to join in the distance at least. I wish I could be in Pisa right now, but unfortunately these are the circumstances. So hopefully in the near future. So as Serena mentioned, I just joined Swansea University in Wales now 1 year ago, but then also COVID-19 came, and which means that I'm still in the process of adapting and building my lab over there. I was 6 years before that at MIT, so most of the things I'm going to present today were actually made while I was at MIT, and I will also talk a little bit of the new direction and my interest in which I'm moving now at Swansea. So as Serena mentioned, I'm going to talk mostly about modeling, but also my idea is how to connect this modeling with the actual experiments and how to actually design materials. So let me give you a bit of background of what I'm most interested when applying this multi-scale modeling. So my main motivation comes from the fact that let's say we are in a world with limited resources, and we have an increasing global population. So that generates, among many other different issues, there are some challenges that are related to climate change, resilient infrastructure, the need for energy sources, the need for food security. But we also generate a lot of biomass waste, and that means that we have different sources of carbon in different ways that we can use actually to tackle these challenges. So my main interest is how do I connect these two? How do I use all this biomass waste to actually face some of the challenges that are connected to this increasing global population and the need for material resources? So within that, what I mostly specialize is what we call materials by design. That means how do we integrate different techniques of modeling, and I'm going to talk a bit more about those techniques that can be applied to any field almost. Basically, density functional theory of quantum mechanics based type of calculations, and molecular dynamics simulations. So how do we integrate this modeling with different synthesis and production techniques for the materials? In particular, I've worked with hydrothermal liquefaction of biomass, but also with silk processing. And there is something in common to all these things, which is that we always have some kind of bio-inspiration. We try to mimic nature, because at the end of the day, any material, any technology we can develop has been already thought and developed by nature. And first thing we need to do is to learn from nature how to solve that challenge. And then that, of course, applies different applications. I'm going to talk about some of them connected to nanocomposites, soft robotics, or redox flow, vanadium redox flow batteries. So let's focus now on this part of the modeling within the materials by design. So the challenge with modeling comes basically because of the length scale and times scale when you are modeling materials. That implies some implicit limitations that make us to work either in the quantum mechanics field, in which we can indeed include electrons in our calculations, and we can, let's say, describe the system very appropriately. But in that scenario, we cannot afford very large systems, and the time scales are also very short. We can then go also into the molecular dynamics. Basically, what we do is we forget about the electrons, and then we say, all right, I'm going to consider that my atoms are just spheres that are interacting with another sphere through some kind of potential, which is, this potential is called force field. So this very simplistic, more, let's say, a classical approach to the description of molecules and materials is what we do for molecular dynamics. Still very accurate, still atomistic, in which we can now afford a bit larger systems, and is very commonly used for the design of proteins, for larger scale nanomaterials, even for concrete, asphalt, and materials of this scale. Now, there are, of course, some other modeling and different scales, mostly continuous models when we work with finite elements. I have not worked on that scale. The biggest challenge in between these continuous models of finite elements or atomistic modeling is this mesoscale, which is some kind of frontier where we are still working on how to bridge this gap into the models. And now what I'm mostly interested in is how can we then accelerate all these by using machine learning techniques. So how can we use deep learning to actually, let's say, speed up the simulations in molecular dynamics as well as electronic structure calculation in quantum mechanics. And then that, of course, is something you don't do in your laptop. So mostly we are working with supercomputing Wales, the supercomputing center of Wales, in which Swansea is one of the partners, as well with Google Cloud, where we can perform already not in our hardware, but in the cloud with Google. So now the other part is by inspiration. So by inspiration, I mean, I've worked while I was at MIT in different projects with many people in which we worked basically trying to mimic nature to produce, for instance, better adhesives, in which we mimic how muscle threads adhere to stones in many different circumstances. To achieve that, we mimic the catechol-rich proteins, modeling a polydopamine catechol-enhanced polymer that have very good adhesive properties. And then we use that polydopamine to actually produce some graphene oxide polydopamine composite, in which we increase the mechanical performance of graphene oxide paper, mimicking the structure of nacre from abalone's seashell. So these are two examples of bio-inspired materials. We also worked on mimicking and producing materials that actually shrink or expand upon changes in pH or temperature. And that was mimicking a protein that is in the jaw of this beautiful sea worm, Nereis virens, We did also the modeling then to model the proteins, model the coordination of metals with the different terminations in the protein. And then also to mimic how is the actual camouflage properties of cuttlefish skin or octopus, where a protein, you have both structural colors and pigmentary colors. So, but there is something in common among all these things that I did while I was at MIT, is that all these materials are designed for performance, which is great. I mean, that was the purpose, and that was what we were looking for. However, in my opinion, this bio-inspiration goes another way. And is that if you think, for instance, in other natural materials like wood, and it applies to any material actually in nature, these are materials that, all right, are complex. They help mitigating climate change. They absorb solar energy. They are mechanically very strong and stable. They are light, but they also can have a lot of ways that actually turns back into the material cycle, and then you get again the material back. So if you think about the overall picture, this buying inspiration is not only about designing materials for performance, but the interesting part, and is where I'm moving now, is how to design for reuse as well as performance. And that goes into the idea of circularity, into circular economy, and how to actually create these circular materials. So that's then my particular vision now is, okay, how do we integrate different building blocks that assemble into a complex material? But how do we design that material in such a way that we already know how this is going to deconstruct back to the initial building blocks? So, of course, that's ambitious, and that's a very big picture. So what I'm doing now is, and I studied also at MIT, is to put some of the pieces needed for that. And I'm basically focused on how to create these building blocks out of biomass, how to then not only incorporate biomass as a building block, but also transform that biomass through hydrothermal processing into different types of carbon nanomaterials, and how do we take also building blocks for those nanomaterials. So all that, of course, then always driven to different applications. I'm basically focusing nowadays into energy storage, bioasphalt, and precision agriculture, and also biomedicine. But the application at the end of the day, and the good thing of this is that you have a playground, which is the multi-scale modeling and the machine learning algorithms, in which at the end of the day, you can tune your material to any application you might think of. So then connecting to that part of biomass, basically is how do we explore all the different types of biomass, and how do we process and model those to actually create all these different types of nanomaterials that they start already to be available, but there is plenty of room still to actually improve and design and synthesize this kind of nanomaterials, or bio-based nanomaterials. So just to give you two examples of the projects in which I worked at MIT, basically we started processing different types of biomass, wood kiting and sewage sludge from wastewater treatment. We apply this hydrothermal processing, high temperature and high pressure. So water is in supercritical water, so it behaves more like a solvent, more like an organic solvent that helps degradation of biomass and repolymerization. And then depending on the conditions, you get mostly three different types of materials. One is up here, it's biocrude oil, a carbon, solid nanostructure carbon, and a solution where you have carbon nanoparticles. So the challenge now for us is actually to model these three, that's in which I'm working right now, and actually how to help improving the processing by the models created in this step. So just as an example, by using chitin from shrimp shells and doing this hydrothermal processing, we can create sand and nitrogen oxygen doped bio-based carbon but of course has nitrogen and oxygen coming from the original chitin. This carbon we use it to decorate some felt electrodes, and then apply those felt electrodes into vanadium redox flow batteries. Basically, what we see is that this nitrogen and oxygen facilitate the absorption of vanadium, and that actually improves also the kinetics. So you can see here that's the chitin-derived activated carbon in the electrode, how it has an electrochemical active surface that is reasonably higher than those from carbon black, taken as a comparison. It actually has a higher wettability, so that you can see here the contact angle of the bio-based carbon from chitin compared to carbon black. And when it comes to cyclic voltammetry, you can see how the actual kinetics for both couples, the 2,3 for vanadium and 4,5, both of them has a better kinetics than those are carbon black, and those also coming from wood. Doing the modeling, what we saw is that actually, indeed, you can compare whether for the vanadium 3 or 2, you can see how all the species with nitrogen has a higher binding energy than those species in which it binds only to carbon. So nitrogen indeed improves the absorption of that vanadium species, and that helps improving the kinetics. The other project I wanted to comment on was also how to then combine these bio-based materials with biomaterials, basically to create conductive films that are biocompatible but also conductive. So what we did in this case is that we process silk coming from silk cocoons. We extract the silk fibrine, and we have a process in which we can create a solution of this silk fibrine. And in this silk fibrine, we put different types of carbon particles coming from biomass. So basically, we explore again wood-based carbons and chitin-based ones. So when you put all these in the solution, we also add calcium chloride to control the humidity and water absorption of the membranes. We can produce these membranes of silk that have this carbon, bio-based carbon embedded. We also made the simulations to actually look at how the silk fibrine domains attach to a surface of carbon with the functionalities that are coming from biomass. You can see how if you compare silk with graphene and then with the bio-based carbons, you see how the absorption for those from chitin are very good. So thanks to the nitrogen, also it actually integrates very well with silk, which is something that doesn't happen with graphene. It was observed actually with graphene oxide, but not with graphene. You can see how the resistivity goes down as you go from pure silk to increasing percentage of chitin-derived bio-based carbon. So it actually becomes more conductive. And then when it comes to mechanical properties, there is an increment also in the failure strain. The actual peak stress and toughness goes up and then goes down as you increase the amount of percentage of bio-based carbon. There is a sweet spot here, which with 0.51, you have indeed lower resistivity, higher failure strain, and higher peak stress and toughness. So you have better mechanical performance and higher conductivity. When it comes also to the modeling, you can see how actually if you compare graphene versus the other two, the changes in secondary structure are way larger for graphene. So also chitin would not only integrate better with silk, but also let's say create less disturbance of silk structure. So these are two of the examples that in which I'm working right now, following up in Swansea. So just to finalize, I think we were running late, so I think I'm good with time now. It was, again, two examples how to move forward in the use of bio-based materials. That the idea, again, the big picture is how to make those circular. So not only reintegrate bio-based, but to design them to go back into something which we can reuse. So let me then finish thanking everyone I've been working with during all this time in this project. So especially to Marcus Buehler, my supervisor for the last 6 years at MIT, and Diego Lopez, who was the postdoc working with me on the daily day and all this, but many others like JJ, Zhao, and many other collaborators that made possible this kind of research. And with all that, thank you, everyone. I'm open to any questions.
Thank you very much, Francisco. So now, especially here in Europe, the bio-based industries, there is a lot going on in this field. So your expertise is very well appreciated. And also there is a lot of space for improvement, and also the application of multi-scale materials modeling is very new, extremely new in this field. So congratulations for your talk, for introducing these new topics of investigation. Maybe there is a question from the other speakers related to modeling or something. This is not my specific field, but if there is some technical question, for example, regarding the types of tools that, because the biological materials are very particular, so it's not very easy to have models, no? So how can you suggest, for example, for future studies in this field and collaboration, so how do you suggest to approach modeling these materials like lignin and chitin?
Well, I mean, for materials like lignin or chitin, I mean, the approach is not that different in principle to those that was followed in the past for protein-based materials. So at the end of the day, because of how these methods were developed, they were made in such a way that we have, let's say, the way of describing how the atoms interact. And because at the end of the day, we keep having carbon, oxygen, nitrogen, and all this, these interactions are already described. The challenge, however, and mostly when it comes to things like lignin, comes actually not that much from the actual method, but from the knowledge we have about lignin in the larger scale, that we need to create a structure for lignin, which is very random, in which we need to have different types of bonds, beta 04, 5, whatever bonds we have there, in a random distribution. And then if we really want to describe this properly, the size of the molecule starts to be really big. And then then that's the challenge, that to describe materials that to really appreciate how they perform, we need to go to certain scales that starts to be a challenge. That's why people is also moving into using machine learning, because the goal at the end of the day is that if you have some algorithm that can predict certain properties without the costly, let's say, atomistic simulations, that could then allow you to go to larger systems by other means. So that's one of the, but I think it's more about, initially it's about following similar path that it has been made in other fields.
So thank you for being here. And if I would like, if you have time, you can stay also online and so synergize also in the discussion with the other speakers of this session. Okay, thank you, Francisco.
Great. Thank you, everyone.
So now we move to the next speaker, Dr. Thomas Stoppe from Dresden Technical University. And so this is also related to some part of simulation of tympanic membrane. And so nice to meet you, Thomas, and you can go with your presentation.
Yeah, hello everyone. I switched to my presentation.
Share screen.
Can you hear me? Is it loud
Okay,
enough?
it's coming. Okay, then you can.
Yeah. Is it loud enough?
Yes.
Can you hear? Can you hear me? Well,
You can put the presentation mode. Okay, perfect. Yes. Thank you.
Okay. Yeah, thank you as well. Yeah, thanks to the other speakers and thanks to the organizing team for this, and mostly Serena for the efforts managing this workshop. Very nice workshop with nice topics. And yeah, I'm going to. Oh, there's something missing. Yeah. Yeah, I wanted to talk about trends in surgical therapy and diagnosis of middle ear diseases. I was asked to focus a little bit more on computation, and so I'm switched kind of like topics a little bit in respect to the abstract. But let me first introduce you to the region of interest. We heard it sometimes before during this workshop. I'm focusing or our research is focusing on the middle ear, which is connecting the inner ear on the left and the outer ear on the right side. A little introduction to the function. So if you've got the dynamic sound pressure coming into the ear canal, it is collected by the tympanic membrane in red here. And the tympanic membrane is then exciting this dynamic vibration in air into mechanical motion of the 3 ossicles. I have a magnified image here on the right side. So tympanic membrane red, then the malleus, then the incus, and then the stapes, which is connected with the footplate on the very left to the cochlea in like bluish or violet color. Yeah, so there's vibration here, this arrow, which we're focusing on in our research. Yeah, I want to give you 2 examples for reconstruction of the middle ear or the tympanic membrane to give a little overview about what's coming afterwards, which I want to focus on. So first, reconstruction of the ossicular chain. So here again, the 3 ossicles, the stapes, the incus, the malleus, and the tympanic membrane. This is an intact middle ear, and if there are chronic diseases or damaging pathological changes like trauma or something, destroying this ossicular chain, the vibration is interrupted, and if the ossicles are destroyed, at the moment we're using rigid reconstructions. So we got, for example, here a titanium prosthesis, which is connecting the tympanic membrane to the stapes here with another small joint, so that vibration can happen. What I forgot to mention is we have this dynamic motion here coming to the canal, but there can also be quasi-static changes. For example, if you're diving or if you're flying in a plane, we have to swallow, for example, to equalize the pressure between the middle ear and the outer ear, or the ambient surroundings. And then the tympanic membrane can move inwards or outwards. So that's what's the problem with this rigid reconstruction here, that this rigid prosthesis it doesn't have joints, so it can't equalize this movement of the blue malleus here, the tympanic membrane. So that's why we thought we have to come up with a joint in the prosthesis. Like usually there are 2 joints from the malleus to the incus, and then from the incus to the stapes. But in birds there is only 1 joint, in the columella, which is called, for example, in the ostrich here on the right side, and it has a single joint middle ear. So if you can see, it's like kind of like bending or tilting downwards and upwards here, if there's ambient pressure, quasi-static ambient pressure. So we developed a silicone-coated ball and socket joint to reconstruct this middle ear with a joint prosthesis, which looks like this in a human middle ear in a preparation. And so if you want to look at the 3D image before, it looks like this. And now we have sound transmission in the same quality as a rigid prosthesis, but we have also the flexibility of an additional joint, which isn't the solution for all the problems, but at least it's a good direction to keep going on. So another thing we are researching is the tympanic membrane reconstruction. Shivish Anand mentioned it before, and other colleagues from mostly Pisa, I think, during this workshop mentioned it, the 4 nanometer project. So if we got, again, chronic diseases, which are not healing spontaneously, like small defects, for example, which are healing spontaneously, example here, if a chronic disease, then we need to, or it needs to be reconstructed with autologous tissue at the moment, like cartilage or fascia or perichondrium. And as you can see, it's not one single patch. There are multiple patches to keep the stability, but also the flexibility for the patient, so that he can hear properly again, and that the reconstruction won't get destroyed after the surgery. Yeah, I think it was also meant for the complex structure of the tympanic membrane is basically inside the collagen layers or collagen fibers. So there are radial and circumferential fibers, different connections, interconnections between each other, and it can be reconstructed with these patches here on the left side with other tissues. So that's why we are focusing on a combined approach of fiber deposition and electrospinning with the guys from Maastricht University, the Merlin Institute, to create on the right side such biomimicking scaffolds with circular and radial fibers. Here in a big picture, and on there are other two scaffolds which we have investigated for determining the results of the radial and the circumferential fibers. I'm not going to show these results here, but as Shivesh asks, because I was shifting the focus a little bit, as I mentioned before, but I can say we have a pretty good sound transfer function of these scaffolds in comparison to the tympanic membranes in the range of kind of like 200 to 400 hertz with the first resonance frequency and about 10 to 15 dB higher magnitude. But we can talk about this in detail if you maybe email me afterwards or ask a question afterwards. Okay, so coming back to the presentation, the topic was about kind of like trends, to give a little broad overview about the middle ear research. There are common trends like digitalization, individualization, bioprinting, rapid prototyping, virtual and augmented reality, artificial intelligence, like Francisco mentioned before, robotics, higher integration, miniaturization, and lots of new materials, which you guys are more experts in than me, I think. And these trends, they can address the unmet needs for autology. So with focus on the middle ear during my presentation here. For example, for the patient, it is required to have an objective and fast diagnostics and therapy. For example, for the implants I mentioned before. So because the surgeon at the moment is reliant on its own expertise in reconstruction approaches. There are various approaches for tympanic membrane or for ossicular chain reconstruction. So if we go back to the example with the ossicular chain reconstruction with the titanium prosthesis, the surgeon reconstructs the ossicular chain and the prosthesis is there and it's in position, but the surgeon doesn't know how it will work out in the end. So he has to wait until the healing process is finished, and then he can do some audiological measurements, and then he will know how the patient will hear and compare to the previous audiological measurements before the surgery. So that's still an unmet need. And yeah, the main target would be to, get a complete new middle ear, for example. So a full biomimicking reconstruction of the ossicular chain and tympanic membrane, or the inner ear, if you go further into the hearing organ. That's still the goal here. And of course, at least for Germany, we need a low price for the treatment, which is not very easy to achieve. As you know, that the medical certifications and so on are getting expensive and more expensive. Okay, so I was talking about computation. Now I'm getting to the topics of this introduction. I want to show you two examples, one more kind of like basic research and one application. Basic research will be about the middle ear morphology, about the shape analysis, which is kind of like relevant if you don't have an individualized implant, for example. So if you can't get 3D data, then you need prosthesis which can fit multiple patients, but not all patients probably. And the other example would be diagnostics. So especially the optical coherence tomography I will talk about. Other related topics during this talk are reconstruction of the middle ears, which I talked about in brief before, and finite element simulations, which you can see a model right here on the top. Osseointegration of titanium implants into the middle ear, measurement techniques For example, here tympanic membrane vibration pattern from at different frequencies, and training models, for example, here on the right, because as I said, the surgeons need to have a lot of experience for the reconstruction. And here we have an artificial model with a plastic stapes and cartilage tympanic membrane from the sheep ears, for example, and the real prosthesis where the physicians can train, for example, during our ear courses in Dresden. So talk about morphology. If you got an implant, you need to know how big is the anatomical variation of your target structure. For example, the prosthesis, how big is the length, for example, or how long can it be, the prosthesis, of course, in 3 dimensions. How much space is there for the implant to fit or to have a certain amount of movement possibilities, and where are the key landmarks of your of the patient, of the different patients, of the variation, anatomical variation. So talking about computation, we took 20 micro-CT data sets to build a statistical shape model, so to get the variation of the middle ear, of the ossicular chain from the human middle ear. And we were vectorizing the variation of the human middle ear morphology this way. And at the end, of course, we want to use this model to investigate the influence on the middle ear sound transfer function in the end simulation model. So, for example, you have 10 stapes, and as you can see, there's a kind of bigger like variation. So the length, the height can be different. The width, the length, the height, the width can be different. There can be certain angles of the crura, of the 2 legs here, and the space between them can be different. And there are a lot of shape variations. The same with the 3 ossicles in combination here on the right side. As you can see, the length of the malleus, for example, can be different, and the position of the stapes foot plate here in kind of like bluish color to the malleus can be different. The angles can be different, and the rotation between the malleus and incus, for example, can be very differently. So we took these micro-CT data sets. Of course, we didn't only have 20. I think we have double the amount, because not all the all the data sets could be used. With a voxel size of 15 to 19 micrometers, that we used no staining for because it brought disturbances in the imaging, and we had a scanning time of about 11 to 15 hours per data set. So with this imaging data, we got an image stack of the tympanic membrane, the malleus incus, the stapes and the inner ear, and this data was segmented within semi-automatic segmentation procedure. We used smoothing, and we got then the binary image and then STL mesh using 3D Slicer and MeshLab. Afterwards, we registered the segmented ossicular chain. We wanted to get a template, for example, here of the stapes. Therefore, we used a mapping with the advanced normalization tool, or shortly ANS, to correlate the single, for example, stapes meshes to one average mesh. And we have to iterate this process over multiple times, because initially we don't know the average template, so we have to correlate the mesh points to the average template, and then we correlate the single stapes again to this template. After the registration, we performed the rigid alignment, so all the correspondences between the single ossicular chains to one another, we put them one over each other, so they were kind of like overlapping. And after a principal component analysis, we got a mean shape of all these data sets and the principal components of the shape change. So, for example, I will show you only the first principal component here. So on the left side, you can see the -3 sigma shape change. So this is not a vibration, but a shape change. So, for example, there was corresponding the +3 sigma and the -3 sigma. So the first principal component was compared to the mean shape in the middle, kind of like this angle of the staypiece to the incus process, and the rotation kind of like a little bit between the malleus head and the incus. Yeah, and we can use this as a statistical model for the simulation in milieu transfer function to later on get the vibration in different patients and with different implants, for example. Okay, going forth to diagnostics real quickly. Yes, I was talking about optical coherence tomography, and we integrated this into an endoscope to get a combination of 3D imaging, so the morphology, and also the functional measurements. So as you can see here on the left, the physician is able to use the endoscope to have a direct view, camera view on the patient's tympanic membrane, and also he can perform functional analysis and get the morphology of the tympanic membrane. You know, the optical coherence tomography, or OCT, it's an acronym, can look through tissue if it's not too thick or too dense. So we can look through the tympanic membrane and see what's behind, and also we can get the functional vibration, which I will show you in a second, in the next slide. So this is the view of the physician, and then we were performing multiple B scans, which are consisting of A scans, and imaging the whole tympanic membrane would be an M scan. Okay, so an M scan is the mapping of the whole tympanic membrane. So we have a diameter of the endoscope of 3.5 millimeters, a working distance of 10 millimeters, and a field of view of about also 10 millimeters. We need post-processing to rectify the image stack and to perform the processing of the functional data and so on. Acquisition time was for an M scan, so for getting the 3D image stack, about 6.4 seconds, and for getting the functional measurements with the interferometer-based approach. As you can see, here's a microphone attached and sound excitation inside, which can be plugged into the patient's ear to get the functional measurements. Took about 4 to 5 seconds. Yeah, for example, here you can, as I showed before, as you have seen before, this is a vibration map of the tympanic membrane of the patient in vitro at different frequencies. So you can see if there would be fluid behind the patient ear, this amplitude would be much lower, and there can be fluid behind certain areas. So certain areas can be inhibited in vibration, and you would see that too. And so you don't need multiple diagnostic steps for the patient, but just this one tool to determine if there's fluid behind the ear and maybe what kind of fluid. Yes, so one last image here about the morphology. So as I said before, we can look through the tissue. So for example, if you have a reconstructed ossicular chain out of titanium, for example, you can see in the cross-section the head plate. So this part here, this round part here, behind the tympanic membrane, and you can still see if it's dislocated or damaged, or if something has grown underneath it, or anything. Fluid here at the arcteryx position. That's what you can determine with this morphology analysis. And you can, of course, get the thickness out of these measurements if you got enough data and knowledge about the technique. Yeah, I want to acknowledge the funding organizations, and although the von Neutron project, and my colleagues Steffen Osman, Matthias Bonitz, and Josef Morgenstern for their work, and I want to thank you for your time. Thank you.
Thank you, Thomas. So the audio was not very good. So, or maybe we need some new tympanic membrane.
Sorry.
Okay, so I think that we could follow your presentation. So thank you for being here, and maybe, I don't know if there is a question, maybe Mario, do you have a question? Or we have a question first from Giacomo Ceccone. This is like he said, maybe something about quantum effects in material difficult to modeling. Do you expect a better chance when the quantum computing will be available? So this is, I think, is a question for everybody in this session. So what do you think, Thomas, about this question? Would you like to have a quantum computer?
Would sounds nice, fancy, but I have no idea about the possibilities about quantum computing. Sorry, I can't answer this
Yeah,
question.
so how much is the computational cost of the simulation, for example? Takes hour, like days, or is it like something that impacts your research capacity?
Yeah, sure. So you know, there's gigabytes of data for the shape analysis as well as for the OCT. And so for the simulation, for example, if you want to run a FE simulation, it can take some hours or maybe some up to 10 days, depending on the resolution you're choosing. And for the shape model, yeah, you need a very good graphics card and a RAM and a fast processor. So also for the OCT, so there are gigabytes of data acquired within seconds. So you need fast data acquisition devices. And yeah, so it's definitely a point to consider in the fast PC.
I have a question for Thomas, if possible. I don't know if we are on time or...
Yes, we can make a question.
Yeah, it's more a curiosity than a real question. Thinking about the machine learning approach, which features of a prosthesis would you like to optimize? I think about topology, but topology is somehow general. But are there specific features of middle ear prosthesis that you would like to optimize, and why?
You know, yeah, you know, the main goal, you have this complex shape of the ossicular chain, right? And for now, with this prosthesis, you have a straight line coupling between the tympanic membrane and the footplate, right? But you don't have this the camera like this shape, you know, the C shape and the stapes bottom. Yeah, it would be interesting to correlate this shape, or why is it developed like this, you know, and what are the advantages in comparison to other joints. Yeah, so this natural shape, where does it come from? That would be interesting. And for prosthesis, of course, if we have a joint, you know, the prosthesis is straight and then it's flapping over, you know, this is maybe this ossicular chain shape, you know, kind of like this direction. What should it look like to have an optimal joint, for example?
Thank you very much.
Thank you too.
Thank you, Mario. So if there is no more question, maybe we can go to the next speaker, but I cannot see her, so maybe I kindly ask the direction if Ina is to put online. Thank you. Hi, Ina. Welcome.
Hello. Thank you.
To the conference. Yes. Yes. So we could not see each other in the Hawaii conference. That would be have been very nice. But anyway, so it's good to see. I created a small group for simulation and in silico. Also, I wanted you as a speaker because you are working in this field. So please, Ina, you can make your presentation. So you just go on the share screen and I wait until it comes out.
Okay.
Share screen, and then you have to press the window that push up the desktop.
It doesn't allow me to share it. Okay.
Then you, click on the desktop window, and then you can find your presentation. Okay, it's coming, and then you put the presentation mode. Okay, thank you very much. You put presentation mode is
Yeah, I think now.
Press. Maybe it can just take time. I don't know. You press it now.
Yeah, press it. I put it into presentation
Try
mode.
again.
Do you see it?
Not yet. Lorenzo. Do you have the presentation button? Oh, it disappeared. Okay. Sì, ma non veniva.
Sorry. Yeah, it's kind of doubling.
Yeah.
Okay.
Yes.
So should I share the entire screen or a specific file?
Share screen and then the desktop window.
Okay.
Then you go on your present like you
I
did
see.
before. Yes.
Entire screen. Okay. And now I'm going to the presentation. Does it work?
Yes. Perfect. Okay.
Perfect.
Okay. Good.
Okay, good. Okay, sorry for technical issues. So my name is Inna Ruganskaya. I'm working as a postdoc at the University of Bremen, Department of Geosciences. And I would like to talk today about a bit unusual topic, but very popular in our field right now. It's biogenic nanomaterials or nanominerals. So everyone is interested in this kind of materials, how they are produced, how do they react in the environment, how do they behave. And I began to make some steps towards modeling this kind of materials. So in this video, you can see a coccolith. It's a marine shell organism, which is basically produces, grows minerals inside in the shape of the discs. And it exerts this disc on the surface in the end, and it's becoming kind of a sphere, which is covered by this shell. It's building its shell in this kind of way. So in general, it's a very interesting question. How do crystals grow inside living organisms, or how do bacteria precipitate minerals? Because geoscience is mostly concerned about kind of inorganic crystallization pathways, and these pathways are already quite complicated, especially at complex natural conditions controlled by different pH conditions, saturation states, concentrations of different chemicals, different ions, electrolytes, and so on and so on. So this question is very important, and it's becoming kind of investigated in more details. And another question is what are reactive properties of this new biogenic or biomimetic nanomaterials? Since they have much more complex structures and textures and different surface behavior, they should react differently. And what is basically biologically, what are the mechanisms controlling mineral dissolution and growth when they interact with the leaf environment? For example, here you can see a model, quite simple, simulating interaction between mineral surface and a bacteria. I think this is Chiavanello. So it's basically introducing some sort of changes into the surface morphology. It's builds kind of a rim around the bacteria itself, and the surface underneath the bacteria dissolves quite intensely. Bacteria produces some acids, it facilitates dissolution. So it's kind of a new player, one more important environmental parameter into mineral fluid interaction in the environment. So this kind of mineral fluid interactions are in general can happen at different scales, as already Francisco said. It's a very kind of complicated process to relate different scales, going from the atomistic molecular scale to the macroscopic world. So I work at the mesoscale, actually. At the mesoscale, I'm trying to connect basically the effects happening at the atomic scale and in the macroscopic world. So in this kind of in-between place, I'm trying to build sort of reactive ensembles, realistic mineral surfaces, some ions in the fluid, and see how do they behave as a whole. So the approach itself, we call it a probabilistic because it operates with the probabilities, and in particular sort of simulate a sequence of the reactive events on the surface, which probabilities are calculated by using environmental parameters plus crystal lattice parameters. I simulate bond break-in, bond forming reactions. They are all activated processes. I need to know my activation barriers to calculate parameters correctly. I need to know saturation state and other environmental controls. So as a result, with the kinetic Monte Carlo simulation, I see surface alteration over time. So here you can see a direct comparison between atomic force macroscopy measurement, this is calcium carbonate dissolving, and kinetic Monte Carlo simulations. I put together different time frames to show how calcium carbonate, mineral calcite, dissolves over time. I have some lattice defects, pretty much screw dislocations that go through the surface and work as sources of the reactive steps on the surface. So in this way, reactive surface area is generated and some certain reactive sites are generated. Going a step further behind structural controls, we also implement fluid controls, in particular ions in the fluid and charged surface groups, which are also very important for the kinetics. So basically, I'm using for this kind of purpose a bit different approach, not kinetic Monte Carlo, which simulates system evolution in time and propagated from one surface configuration to the other. I simulate a reactive ensemble as a whole and generate a large number of possible combinations, surface and surface groups plus ions in the fluid to get the statistical behavior of this kind of system and relate it further with the kinetic Monte Carlo models, with the kinetics. So this kind of equilibrium grand canonical Monte Carlo simulations dealing with statistical ensembles plus kinetic Monte Carlo simulations dealing with the reactive events are put together. Right now I'm working with magnesium calcium carbonate solid solutions. This is important biomineral of marine organisms. And these kind of materials are becoming much more interesting and complicated than pure phases. Why is that? Because when do we add a different kind of cations into the system, we can generate a large possibility of arrangements for the surface bonds, and each carbonate ion can get a different coordination with respect to magnesium CO3 bond, calcium CO3 bond as well, and this kind of two bonds have very different activation barriers or rates to break. So pure phases, pure calcite, calcium carbonate, and pure magnesite, magnesium carbonate, have 3 orders of magnitude difference in solution rates. So their behavior is very different. And the first idea about dissolution behavior of solid solutions is pretty much about different rates of breaking for these bonds, but there is another important control which people became aware about. Since this kind of cations have smaller size, they introduce lots of the strain into the lattice. So experimental observations for growth of this kind of material showed that it cannot grow smoothly, actually. It forms sort of segments, and this kind of segments, arrangement, and formation is controlled by the internal lattice strain. And related observations, experimental observations, pretty much showed that dissolution of this solid solutions is faster and solubility is lower than pure phases. So why is it? Pretty much mismatch between the cation size causes displacement within the lattice, and the displacement causes strain. So the lattice is internally strained, and this strain induces excessive energy, and which means that the bonds are in general breaking faster than the pure phases. So say if we introduce lots of substituting cation into the material, the entire lattice becoming strained. And in general, strain energy density can be calculated. I did it for calcium carbonate, and it can be calculated by using strain tensors and other computational approaches, and all this kind of information for the strain energy density comes into the equation for the dissolution rate. So in this way, we can understand the role of the strain. For example, with solid solution materials, 20-40% surface morphology changes. It is also in a bit different way. And this kind of contribution of the strain is substantial for every atom. Every atom is surrounded by a large number of sort of strain centers, such as magnesium, and this kind of effect for this kind of material is actually very strong. It takes place within 20 nanometers radius. So it means that every atom is strained, and for every atom I have to calculate big strain tensors for the entire lattice. It's becoming kind of computationally intense. So this is a preliminary results for the dissolution rates. I took at this stage a bit small system and try to understand how fast this mineral will dissolve if I dope it with a different amount of magnesium. So the result is incredibly interesting. It showed that material can dissolve up to 5 or 6 orders of magnitudes higher versus pure material itself. But if we add a small amount of magnesium, such as 5 or 10%, the rate is not really different. So in general, we know already that these materials are very reactive due to the strain. Biogenic carbonates contain typically up to 20% of magnesium everywhere, and this is quite interesting, and even up to 40% in some parts of the body. So what are the evolutionary reasons for this kind of selectivity for the solid solutions? We know in general that they have kind of enhanced elastic properties, so they kind of more kind of tense, and it's kind of harder to destroy them mechanically, but chemically they obviously easy to destroy. And so what are the mechanisms of the mineral retention in this case within these organisms? And another interesting problem, I think, which is kind of closely related with the problem of solid solution behavior, is human bone mineralization or resorption. Major mineral in the human bones is hydroxyapatite, in particular carbonated hydroxyapatite. It means it's solid solution again, but here orthophosphate ion is substituted by calcium carbonate ion, and this kind of change introduces some strain and definitely charge vacancies which are present. So crystallization in many experiments show is driven by the surface charge. It's the organic complex assembly, whatever it is gelatin, collagen, whatever, which is basically generates some kind of surface charge, and the surface charge electrostatically attracts the material, pretty much calcium and phosphate, and the material starts to grow. So interesting problems to explore in my case. Generally, what are the mechanisms, molecular mechanisms and mesoscale mechanisms for the growth this kind of material, what is the role of the strain and the surface charge, how it grows or dissolves under the influence of the strain and the charge, and how these synthetic materials, for example, can be produced and how they will behave in vivo, in the natural environments. This kind of questions have some practical interest, but also very interesting fundamentally. I think it will explain us how these minerals can behave. So in general, the Monte Carlo methods can be used as a mesoscale model that can help us to understand growth and dissolution of biominerals atom by atom, their reactive properties, and biologically controlled dissolution. So next, I think we still have a big room to study mechanisms of this kind of living organism and the mineral interaction and the mechanisms of growth. And we definitely need for building mesoscale model some relevant key kinetic parameters and rates of these processes and key reactions. And it would be interesting to explore such an area as bioengineering for production of this kind of environmentally relevant nanomaterials. So I would like to acknowledge all my colleagues and friends at 4 different universities where I worked and studied. Of course, I could not build my knowledge and expertise without them. This is mostly my advisors and colleagues from the University of Bremen and Marum, and Rice University, where I did my PhD, University of Bern, where I worked, and colleagues from Paul Shore Institute in Switzerland. Thank you. And I will be happy to answer your questions.
This was a very interesting presentation. There are in this conference many scientists interested in the biomineralization, like tissue engineers, and so I think they will appreciate like more tools for predicting the behavior of their scaffold or materials. So this is, I think, a new topic for tissue engineers to include relevant tools for simulation. And so my question is if these tools can be applied also for like a complex system, like in vivo, for example, or in vitro. Is there any any way to include more like information in your model?
That's a very good question. There is no limitation in general for the complexity. We can add as many processes as we want when we're limited by the memory size on the machines. But we have to be very careful. So we should select the most important processes, basically the processes which controls rate limiting steps, and the fastest processes. They're also very important. So the model itself is probabilistic, and we are playing in the field of probabilities. So it depends how do we define this kind of set of the probabilities. If we will do this correctly, if this will mimic real kind of physical picture, then yes, we can model this. So it's not so much about capability of the model itself, it's about our knowledge about the key, the relevant processes, but all other physical information, charges and ions and rates and type of reactions that can be added, but probably it will require some kind of coarse graining. It would be very difficult to simulate every proton, every whatever sodium ion, every electron. In general, it can be done, but it's kind of makes calculations very, very inefficient. But we can think towards that. It's a very interesting question. We're very interested in thinking in this direction.
Okay, thank you. So maybe the co-speakers in your session, do they have questions, Francisco and Mario? If you don't have question, maybe we move to the last presentation of Dr. Mario Milazzo. Is there any question? No? So we move to the final presentation of this conference. And it is my great pleasure and honor to introduce Dr. Mario Milazzo, so from MIT and also Scuola Superiore Sant'Anna. And he will conclude, I think, in the best way, this very successful conference with his talk. Please, Mario, you can have the speech.
Thank you very much, Serena. Can you see my screen?
Yes.
Okay, if you can see the full screen, I can start. So thank you all for being here. I'm the last one, so I really appreciate if you're able to listen to my presentation. Thank you very much, Serena, for the nice introduction. I'm Mario Milazzo. I'm a postdoc researcher at the Department of Civil and Environmental Engineering at Massachusetts Institute of Technology, but I'm also a postdoc at the Biorobotic Institute at Scuola Superiore Sant'Anna. I'm going to present you a topic, the work that I've been doing the last 2 years. So I've been studying the viscoelasticity properties of collagen-based materials using molecular dynamics approaches. So somehow I'm using one of the methods that Francisco introduced in his presentation. Why is it so important to understand the viscoelastic properties of collagen-based materials? First of all, because collagen is at the base of many biological materials, such as muscle and tendons, or speaking about the ear, we already listened to Thomas Toppe that spoke about the ear structure. So somehow I want to also include some hints also in this topic. So collagen is at the base of the tympanic membrane, in which collagen fibers arranged here, you can see in radial, circumferential, and parabolic arrangements. They allow the transmission of the acoustic signal through the middle ear, and but also because collagen is at the base of the bone. So if we go into the deep and we go at the atomistic scale of bone, we will find the hydroxyapatite, but collagen, and also water. So my question is there a way to better understand fracture or bone conduction, so a different way to transmit acoustic signals to the brain, or design new materials based on bone, understanding the viscoelastic properties of bone itself? Because in this way, we can better understand physiology, but also we can design, let's say, super materials in which we can exploit the benefits of having specific characteristic of the bone, but avoiding at the same time, let's say, the disadvantages of the bone itself, creating therefore somehow the better material than the physiological ones. So starting from the beginning, my study started studying the collagen peptides. So just take into account little stripes of peptides, of collagen peptides. So you can see we studied dry and hydrated structure with different loads in order to understand either the hydration or the loading directionality, how these factors can affect viscoelastic properties of the collagen. So we studied 2 cases in which you can see a modeling, like we model the collagen as a rod, in which we stress the rod with longitudinal waves, impulsive waves, you can see here, or modeling collagen as a string. So we pre-stress the collagen, and then we stress the collagen with a transversal load, and you can see here the deformation like a string. The modeling itself uses very simple equations to understand the mechanical properties of collagen. So for longitudinal waves, we are using the classical relation between velocity and Young's modulus and density, while for transversal waves, we use the model of a string. So we use the tension of the string, and therefore we can relate the Young's modulus with the pre-deformation epsilon of the string. At the same time, we can understand not only the transmission of the waves, the transmission of the energy, but also the dissipation of the energy. And in this case, we study the square value of the velocity of the front wave in order to understand how fast waves are dissipated through the structure. Few plots. What I want to show you are mostly plots, not numbers, just to understand the phenomenon. So in this case, we are looking at the plots for the longitudinal case, in which we compare the dry structure with the hydrated structure, and you can see that the plots are basically the same, unless you can see here a slope for the propagation of the wave that is a bit steeper for the hydrated structure, and this means that there's a different velocity between the dry and the hydrated structure. Specifically, we can see that the velocity of the waves through the dry structure is much faster. So we are about 300 meters per second, and this corresponds to a high gas modulus, while for the hydrated structure, interestingly, we see a slow velocity, relatively slow, and the less stiff material and the less stiff Young's modulus. As for the dissipation, since also the plots are showing us basically the same representation of the situation, we can see that basically water is not affecting significantly the dissipation behavior of the structure. A different situation is here, looking at the collagen modeled as a string. In this case, we pre-stress the collagen, as you can see also in the video, and then we apply the transversal load. So on the left, you can see the panels related to the dry structure, while on the right, you can see the plots related to the hydrated structure. By pre-stressing the peptide, you can see here 1% and 10% for both cases. You can see as for the dry structure, we have an increase of the frequency related to wave transmission, because the structure is becoming stiffer. While looking at the hydrated structure, we cannot see any specific difference between 1% and 10% of the pre-strain. This means that water is really affecting the behavior of the structure, and is significantly dissipating energy. But let's increase now the complexity of the model. I showed you just collagen by itself. Now let's see collagen that is mineralized. So at the bottom, you can see only collagen without any percentage of mineralization, and specifically for mineralization, I mean a percentage of hydroxyapatite inside the fibrils of the collagen fibrils. And the other two features, the one in the middle is collagen fibrils with 20% of hydroxyapatite, and the last one is 40% hydroxyapatite. These are the three topologies that I studied in order to understand how waves propagate inside this kind of structure. Inside this kind of structures. In order to simplify my model, of course, I used only this section for the three topologies. So I studied only the D period composed by the overlap and the gap region. And you can see in the gap region, there's the hydroxyapatite is mostly accumulated. So we expect also before the simulations that this region is much stiffer than the other one. In this case, we stress the structure only with compressive or tensile loads with different input velocities. And in order also to understand if a different velocity, a faster input velocity or a slower input velocity can change the behavior of the material itself. And here, before showing you all the plots, here's a nice video in which I can show you qualitatively, because of course waves are not sinusoidal, but it's just a qualitative simulation of what's going on. You can see that there's a traveling wave starting from where the load is applied, and when the wave hits the interface between the two regions, the overlap and the gap region, we have a reflection of the wave. I can show you again. There's a reflection of the wave that creates a simultaneous rebound of the waves. This is really interesting because you can see it in both compressive load cases and the tensile load cases. Here, there are a lot of plots. What I want you to focus on is just this. So each plot concerns the topologies with 0, 20, and 40% of hydroxyapatite with an increased input velocity for both compressive loads and tensile loads. You can see that the plots are somehow similar, but somehow different. First of all, we can see that increasing the input velocity for both compressive loads and tensile loads, we have an increase of the dissipative behavior. And this is something that we will see later mathematically also. And this is also something that is really connected also to the macro scale and how bone works, because if we eat our bones with very fast loads, bone structures are able to dissipate very fastly energy, avoiding any damage on the structure itself. Then another nice thing to see is the change of slope that is between the overlap and the gap region. And we can see it better here. And therefore, we can understand separately the velocity of the waves in the overlap and in the gap region. And we can model the wave transmission in both regions separately. And this is, up to our knowledge, the first study in which scientists did this. As for the modeling, also here very simple equations. So since the model is basically a beam, we are using always the same relation to understand and estimate the Young's modulus compared against the wave velocity and the density of each region. And we can create some functions that are averaging the velocities that we are measuring in the overlap and in the gap region by using specific average functions, as you can see. And we can do this for the velocity and for the Young's modulus. We are always interested also in understanding the dissipation of this energy that is transmitted through the structure. And we are using basically the same approach that I showed you before. So we are studying how the kinetic energy represented by the maximum velocity of the front. Of course, we are using the square because it's the energy. And we are fitting this energy with a decreasing exponential function in order to understand the relaxation time that will be our measurement for the dissipation. General plots in order to understand how the material works upon impulsive loads. So what we can see is this. Increasing the input velocity, we have a decrease of the wave speed and a decrease of the Young's modulus. This is an apparent contrast with other simulations that can be done on single peptides, in which if we are stressing with impulsive loads, we are increasing the Young's modulus. But in this case, we are not considering only a single peptide, but we are considering a complex topology in which multiple peptides are connected through hydrogen bonds, and those hydrogen bonds will play a significant role in transmitting energy and dissipating energy also. And this is what I want to show you in this slide, because looking at the relaxation time measurement, we can see that, as I anticipated you looking at the plots, increasing the input velocity, we have a decrease of the relaxation time. So the material is able to fastly dissipate energy. But we cannot appreciate any difference between tensile and compressive loads just based on the plots that I showed you, even if we are using complex mathematical tools. This is why we opted for looking at the 8 hydrogen bonds. And by the number of hydrogen bonds, after the first region in which we are applying the load, we can see that basically the order of magnitude of hydrogen bonds is the same for pushing and pulling loads, as I showed you before, but we can see that there is a slight difference, and compressive loads are, let's say, they give life to a more dissipative behavior, but the difference is almost negligible. But if we want to distinguish between the two cases, this is a good way to do it, looking at the hydrogen bonds. The last step that I want to show you is adding the last component of mineralized fibrils that are composing bone. So we are adding water in this case, ionized water. So we are adding water to the voids of the 0% hydroxyapatite. That means a material that has only collagen, so it's pure collagen. But we are also adding water to the 20% hydroxyapatite, so a material that has 80% collagen and 20% hydroxyapatite, but we cannot add water to the 40% hydroxyapatite because our approach modeling collagen fibrils in this specific case, with this specific topologies, our approach is to fill all the voids with hydroxyapatite. So if we are filling all the voids with hydroxyapatite, we cannot add other water. Of course, if we want to add water to, let's say, to highly mineralized collagen fibrils, we should use another topology in order to also include the interfibrillar water. In this case, we use two methodologies. The first methodology is a dynamic mechanical analysis, the famous DMA, but we use the angular DMA because the extensional DMA was impossible to pursue due to the stiffness of the collagen stripes, and when we were measuring our outcomes, that I will speak a bit in a moment, when we were extracting the results, basically we were looking at the stiffness of the stripes instead of the stiffness of the topology itself. So we used two different angular deformations, and we applied a cyclic angular load on the structure in order to understand instead let's say the behavior along the main axis, along the x-axis. We use the same approaches that I showed you before. So an impulsive load at one end, fixing the other end of the topology. I know that these are complicated plots, but just follow me in a few remarks. So the first remark is related to the quantities that I'm showing you. So G prime, G second, tan delta. So the first one is related to the elastic behavior of the material. The G second is related to the viscous behavior of the material. Tan delta is the ratio between the two, and is another way to emphasize. It's a way to emphasize the viscous behavior of the material itself. So we are showing with the solid lines the behavior of the material with the highest angular deformation, and with the other lines just the smaller angular deformation. In both cases, we can see obviously that increasing the percentage of the mineral, we can increase the elastic behavior of the material itself. Of course, the material is getting stiffer. The very interesting part is related to the viscous behavior of the material, because the viscous behavior doesn't show any specific trend with the lowest angular deformation. Here is summarized by this line that is statistically a statistical manipulation of this data, we can see that basically we don't have any trend, but we can see a specific trend for the viscous behavior against the highest angular deformation, especially after 5 gigahertz. And it's quite linear, and here is represented by this green line. The same behavior, as I was saying, is emphasized in the tan delta. It's possible to be appreciated here in this last plot. Now, adding water, what happened? What happened when we added water to the compound, because we saw something related only to the dry fibrils. So adding water, we are not seeing any specific change for the elastic component besides a progressive saturation that is below 3 gigahertz. Instead, the viscous behavior is significantly affected, and you can see that especially at high frequencies we can reach very high values. The same looking at the lowest angular deformation, we can see a almost similar scenario in which we have a saturation of the elastic component towards the high frequencies, while the dry structure always have let's say an almost constant value. And water, of course, is increasing the viscous behavior of the material. This is not surprising, but this is a way to actually quantify this effect. As for the longitudinal waves, I'm just showing these tables, but what I want to show you is the difference between the wave speed in case of the dry structure and the wet structure. We have an average increase, a significant increase of the wave speed when we add water. So water is enhancing wave transmission, energy transmission, but at the same time is enhancing the dissipative behavior also along the main axis of the collagen fibril. And we can see that there's a ratio of from between the 10% and the 20% between the dry and the wet structures. So the final remarks, just to summarize this journey that I wanted to show you, these are the main remarks, the main takeaways. So impulsive loads are an efficient tool to estimate material properties, especially stiffness, the dissipative behavior of structures, especially for biomaterials, in which we are really interested in understanding this kind of behavior. The second thing is collagen plays a very important role in transmitting and anticipating energy across structures. Collagen is the main actor in dissipating energy, especially because when we are speaking about mineralized fibrils, hydroxyapatite, of course, is a very stiff material, so collagen is the one that is playing the main role, together with water that is promoting the dissipative behavior, is enhancing the behavior that is persistent promoted by collagen, while hydroxyapatite contributes to energy transmission since it is a stiff material, so it's giving, let's say, a boost to energy transmission, but doesn't play a significant role in dissipating energy. At this stage, I would like to thank my labs, my lab at Massachusetts Institute of Technology, my lab at Scuola Superiore Sant'Anna, and my collaborators at University of Pisa. So thank you very much.
Thanks, Mario, for your presentation. It is very interesting. I know your work. I wanted to know if there is a question from one of the co-speakers in this session. Otherwise, I would like you to briefly explain the potential of these results. So there are several tissues and organs, and then understanding this behavior, how they work under this condition, can bring new advancement also for regeneration and therapy. So if you want to briefly comment on this, would be nice.
So for sure, understanding specific properties of biological material will help us understanding the physiology of the material itself, but also creating new structure, new bio-inspired materials that can be used for tissue regeneration, especially for creating scaffolds, for example, scaffolds in which we can culture cells and create replacements for damaged tissues. I spoke about bone, but I spoke about pure collagen. So in this case, we can think about replacements for example, the tympanic membrane, but the bone itself, because it's true that, let's say, bone replacements are, let's say, now already treated in literature very, very deeply, but somehow we are missing some points at the bottom, because some biological structures are always giving us new, let's call them surprises. We are not getting the basic principles somehow, and a better understanding of the material at the smallest scales can really help understanding better materials, understand better how tissues can host these new materials, and what are the effects, let's say, in the long term, without any problems, any rejections. Rejections are, let's say, the most critical issues when we are speaking about tissue regeneration and replacements.
Propagation is very important because contributes to healing. So it is very important to understand the physiology in order to have truly biomimetic substitute. No, so this is a very important topic still needs to be fully understood. So I wanted to thank you, Mario, first, to be here, and also the other speakers and all the speakers that were here in the auditorium these 2 days. It was very intense, very full of really nice lecture, and I wanted to close the conference from here, but room B is still going, so I would just say bye from this podium, and I will move myself to the room B, where the last speaker is speaking, and then I will officially close the conference from room B. So if you like, you can you have the link for the YouTube, and we can officially say goodbye from the other room. Thank you very much.
Thank you.
Thank you.
Thank you.